Hi guys, I'm going to try to read this article, and it's from the U.S. National Library of Medicine, National Institutes of Health. Sorry. Okay, I'm going to see if I can read it. RepA-WH1, the agent of amyloid proteinopathy in bacteria, builds oligomeric pores through lipid vesicles. Let's see if I can do this without getting tongue-tied. RepA-WH1 is disease-unrelated protein that recapitulates in bacteria key aspects of human amyloid proteinopathies. It undergoes ligand-promoted amyloid. This is when it tongue-ties me. Amyloidogenesis. Its aggregates are able to seed or template amyloidoses on soluble protein molecules. Its conformation is modulated by heat shock protein 70 chaperones in vivo, generating transmissible amyloid strains. It causes proliferative senescence. Membrane disruption by amyloidogenic oligomers has been found for most proteins causing human neurodegenerative diseases. Here we report that for PRP prion and synuclein acidic phospholipids, and also promote RepA-WH1 amyloidogenesis in vitro. RepA-WH1 molecules bind to liposomes where the protein assembles oligomeric membrane pores. Fluorescent tracer molecules entrapped in the lumen of the vesicles leak through those pores, and RepA-WH1 can then form large aggregates on the surface of vesicles without inducing their lysis. These findings prove that it's feasible to generate in vitro a synthetic proteinopathy with a minimal set of cytomimetic components, and support the view that cell membranes are primary targets in protein amyloidosis. A long-standing issue on human amyloidosis is the identity of the primary cellular target for the protein aggregates that lead to disease. A number of different, often interwoven pathways have been described as involved in the etiology of most neurodegenerative proteinopathies, including gain of toxicity, phenotypes such as mitochondrial damage with generation of reactive oxygen species, or the aggregation and sequestering of essential cell factors. However, direct damage to the cell membrane is the mechanism most commonly found when amyloidoses are addressed in vitro. In the studies on the interaction between amyloid aggregates or fibers with reconstituted cytomimetic lipid vesicles, liposomes, such as large or giant unilamellar vesicles, LUVs and GUVs, respectively, these findings, hold on. Sorry, I'm not. Okay, these findings are compatible with other gathered from the incubation of amyloid aggregates with cultured cells, thus assessing their toxicity. Nowadays, it's accepted that membrane targeting is an early route towards amyloid cytotoxicity and cell death. Bacteria are prokaryotic microorganisms naturally freed of amyloid diseases although they use extracellular secreted amyloids as functional devices to scaffold biofilms or to regulate the activity of antibiotic peptides. Within a synthetic biology framework, we have recently undertaken the bottom-up design of a model amyloid proteinopathy in the bacterium E. coli. In a first stage, we engineered RepA, a protein coded by plasmid PPS10 by deleting the C-terminal WH2, thus leaving the N-terminal winged helix domain. WH1. This decoupled DNA promoted conformational transitions in RepA-WH1 from its natural role, enabling RepA as DNA replication initiator. After incorporating in RepA-WH1 a mutation, A31V, known to enhance protein-to-protein interactions, expanding the DNA replication capabilities of PPS10. A DNA-modulated amyloidogenic part was generated and treated successfully in vitro. RepA-WH1 thus shares the mammalian prion protein, the ability of nucleic acid ligands to promote amyloidogenesis. In the second step, RepA-WH1 was fused at the C-terminus to a fluorescent protein, M-cherry, thus replacing the original WH1 domain in RepA. When expressed in E. coli, this protein fusion first assembles amyloid precursors in the nucleoid, which then are excluded to the cytoplasm. RepA-WH1 aggregates elicit a proteinopathy, as become evident in time-lapse microscopy studies showing a decrease in the proliferative fitness, significant increase in generation time. Sorry, I have to have some water. I apologize. I'm not feeling well, so I'm kind of a little shaky too as I try to read this. Okay. Let's see. In the second step, RepA was fused at the C-terminus. Did I get there? RepA-WH1, I'm probably going back some. Aggregates elicit a proteinopathy, as become evident in time-lapse microscopy studies showing a decrease in the proliferative fitness, a significant increase in the generation time of the cells, a phenotype linked to aging in bacteria. Cytoplasmic RepA-WH1 aggregates were able to template their amyloid conformation on soluble protein molecules in vitro and in vivo, a hallmark of the molecular transmissibility of prions. Although the RepA-WH1 aggregates are vertically inherited from mother to daughter cells, they lack horizontal microbiologic infectivity. Thus, they can be regarded as prionoid, which, together with its lack of significant sequence similarity to any mammalian protein, qualify RepA-WH1 as biosafe. Subsequent microfluidic studies in vivo revealed that the RepA-WH1 propagates as two distinct conformational variants or strains, showing different aggregation morphologies and degrees of toxicity, which are epigenetically inherited by the bacterial offspring among generations. DNAK, the heat shock protein 70 chaperone in E. coli, was identified as the single cell factor modulating the switch to the variant with a lower toxicity and enabling the generation transmissible particles. Overall, the RepA-WH1 Prionoid matches many of the features of clinically relevant amyloid proteinopathies, providing a simple molecular platform to address in bacteria the complexity of protein amyloidosis. In this work, we have explored the interaction between RepA-WH1 and vesicles of different sizes, and which is LUVs and GUVs, and phospholipid PL composition. Incubation with vesicles either resembling E. coli cell membrane or including PLs with acidic polar heads, APLs, phosphatidylglycerol, and cardiolipin, promoted the assembly of RepA-WH1. I have to have water again. As pre-amyloid oligomers and fibrils. Furthermore, RepA-WH1 binding to LUVs and GUVs released a fluorescent tracer, calcium, pre-confined in the vesicles, thus enabling to follow the kinetics of membrane leakage by means of fluorescent spectroscopy. LUVs or microscopy, GUVs, RepA-WH1 dimmers. I wish there was a different thing. RepA, RepA-WH1 dimmers were more efficient in targeting membranes than performed aggregates of the protein. Membrane disruption in GUVs did not result in lysis of the vesicles, suggesting the assembly of discrete oligomeric protein pores by RepA-WH1, which were visualized by transmission electron microscopy 10. These in vitro assays allowed testing a number of natural polyphenolic compounds known to counteract the amyloidoses of proteins involved in human disease, in common with the mammalian prion protein having both nucleic acids and APLs as effectors of amyloidosis, qualifies the prionoid RepA-WH1. So it's a prionoid, this RepA-WH1, a robust proxy to model human amyloid proteinopathies through minimalist approaches, either in bacteria in vivo or as reported here in cytomimetic assays in vitro. The results. Binding to the internal membrane or acidic phospholipid vesicles promotes RepA-WH1 aggregation. Okay. Previously published results indicate that binding of the bacteria RepA-WH1 Prionoid, especially of its point mutant A31V to different double-stranded DNA sequences, leads to the assembly of the protein into the distinct amyloid nanostructures, including their oligomers, spherical aggregates of various sizes or well-ordered fibers. Inspired by the fact that together with RNA, phospholipids are key cofactors of the prion protein replication in vitro, we decided to explore if PLs could also have a role in RepA-WH1 amyloidogenesis. We studied the interaction between the hyperamyloidogenic variant RepA-WH1 A31V fused to the fluorescent reporter mCherry because this chimeric protein had been defined as the most cytotoxic in vivo, and the internal membrane fraction of E. coli, since the host where the RepA-WH1 proteinopathy had been characterized. Since this is the host where the RepA-WH1 proteinopathy had been characterized. In these vitro assays, membrane preparations surpassed their most efficient effector DNA sequence in getting visible aggregation of the electron microscope, i.e. from less than 20 days, OPSP double-stranded DNA to barely 2-hour membrane, or is that 2? I guess that's hour, and relieved the need for a crowding agent in the reaction. See methods. Sorry, my mouth is getting so dry. Instead of the highly ordered long and thick straight amyloid fibers as for RepA-WH1, shorter, curved, and thinner photofibrils were obtained for the protein fused to mCherry. As the same intermembrane preparations were efficient in promoting the assembly of the standard mature RepA-WH1 A31V multifilament fibers. The most likely explanation is that the C-terminally fused mCherry protein would be imposing steric constraints on the lateral assembly of the photofibrils into the nature fibers. In parallel, as controls, we studied the association states and the secondary structures of RepA-WH1 A31V mCherry, either in the presence or in the absence of a his N-terminal tag and of isolated his mCherry. The latter was monomeric and histidine tag did not alter the association state or the structure of the protein, although the unfused RepA-WH1A31V was dimeric. RepA-WH1A31V M cherry included, besides dimers, a significant aggregation or aggregated fraction, as shown by the dispersion of the sediment coefficients towards higher values, attributable to the presence of oligomers. CD spectroscopy revealed that the fusions were thermally stable values, albeit not matching the extreme stability of their parent RepA-WH1, suggesting that some destabilization of this domain by the C-terminal in M cherry. The spectra of the individual components in the fusion were additive, and their algebraic addition nearly matched the spectrum of the whole protein, indicating that RepA-WH1 and M cherry were essentially independent folding molecules. Oh, goodness gracious. Amyloid seeds. They're learning to build amyloid seeds. They've been using this bacteria. A commonly proposed mechanism for amyloid cytotoxicity implies binding and leakage of cell membranes. The interaction of RepA-WH1 with suspensions of large unilamellar vesicles, LUVs, with various lipid compositions was assayed. LUVs were appropriate probes for getting kinetic insight into membrane targeting by amyloids because, due to their small radii, they exhibit reduced light scattering and have a large surface curvature, thus being sensitive to protein insertion into the lipid bilayer. In these assays, LUVs bore the fluorophore calcium encapsulated. Fluorescence emission increases in calcium is released into the medium, thus acting as a probe for membrane leakage. The lipid composition that led to the maximum fluorescent emission upon incubation with RepA-WH1 included mixtures with an optimum around 50%. The zwittertonic phospholipid POPC, in short PC, with acidic phospholipids, APLs, with negatively charged polar heads such as POPG, PG, or cardiolipin CL, higher fractions of the negatively charged PLs resulted in intrinsic destabilization of the vesicles, leading to some more leakage induced even by M cherry. Time course measurements leakage showed that RepA-WH1 A31V M cherry, either with or without his tag, damaged the vesicles readily, exhibiting a logarithmic release profile, whereas RepA-WH1 A31V control without the M cherry moiety showed a nearly linear plot, and M cherry barely had any fluorescence releasing activity by itself. The observed differences also suggest that the his tag enhances binding affinity of the RepA-WH1 A31V M cherry to lipids at the mild acidic pH of the experiments at 6. The protonated imidazoles of the 6 histidines should increase the electrostatic attraction of the protein for APLs. However, the his tag had not a major role in vesicle leakage because, as noted above, his M cherry was inert in the calcium release assay. In the absence of the C-terminal fusion of M cherry, RepA-WH1A31V had its capacity to leak phospholipid vesicles reduced. This could be attributed to a higher rate of amyloidogenesis for the chimera with the fluorescent protein reporter, as suggested by its lower stability. Alternatively, since the fusion to M cherry led to a net increase in the fraction of oligomerized sorry, I have to. RepA-WH1 A31V especially for the his-tagged variant. Oligomers might have higher activity on membranes than dimers. To address this possibility, the dimeric and oligomeric components in RepA-WH1 A31V mCherry preparations, I'm sorry, this just gets repeating it over and over, fractionated by gel filtration and analyzed through inline multi-angle light scattering. Both fractions were assayed for their ability to leak calcium encapsulated in LUVs made of 1-to-1 mixtures of PC and PG, as indicated above. Interestingly, when assayed at equal concentrations, the chimeric protein fraction was twice as efficient as the oligomeric one in promoting calcium release. The findings suggest that binding of the dimeric RepA-WH1 to the membrane must be directly linked to the event of vesicle leakage, whereas the protein aggregates generated in the absence of effector lipids are less competent to bind and or leak membranes. To get further insight into the interaction between RepA-WH1 and the membranes, we monitored the interaction between the prionoid and giant unilamellar vesicles, the GUVs, made of different lipid compositions, and using diverse assembly procedures. Vesicles were visualized by means of a confocal laser fluorescence microscopy in comparison with the control purified mCherry, which remained as diffused fluorescence soluble protein in the lumen of the vesicles. RepA-WH1 A31V mCherry became visible as protein aggregates on the surface of the GUVs when the vesicles were prepared with purified bacterial internal membranes. This observation was repeated with GUVs made of PLs with the average composition characteristic of E. coli inner membrane. Aggregation of the surface of the vesicles was also evident less complex PLs compositions, i.e., if GUVs included APLs, PG or CL, it is noteworthy that their concentrations of APLs, less than 50%, would not be reportedly tested due to their destabilizing effects on GUVs. However, RepA-WH1 A31V mCherry remained soluble, as the mCherry control did if the lipids were exclusively neutral, PC. The inference from the LUVs and GUVs minimal model membranes is that APLs present in the E. coli internal membrane act as cofactors promoting RepA-WH1 amyloidogenesis. In order to require if the lipid-promoted RepA-WH1 A31V aggregates had amyloidogenic nature, that word still, it's tough for me to say. We probed the protein-bound GUVs by means of immunocytochemistry, an antibody specific for RepA-WH1 oligomers on pathway towards the assembly of amyloid fibers, bound to the aggregates made by the hyperamyloidogenic protein RepA-WH1 A31V mCherry. This antibody also recognized the aggregates assembled by RepA-WH1 WT mCherry, an otherwise soluble, mildly amyloidogenic variant of the prionoid. When this protein was clustered and thus forced to aggregate on the membrane by coordinating its N-terminal his tag with the however you say that, to second our activated polar head of a chelating lipid, DOGS NTA, or DOGS NTA. On the contrary, mCherry was not bound either by the B3B7 antibody or by a WH1. A conformative, unspecific, I have to move around my legs going. On the contrary, mCherry was not bound by either a conformation unspecific anti-RepA-WH1 polyclonal antibody. Overall, the results shown in this section are comparable with the enhanced pro-amyloidogenic aggregation of RepA-WH1 upon its binding to lipid vesicles. To achieve high single vesicle resolution in the leakage assays, we incubated the different protein constructs with GUVs in calcium and encapsulated in calcium, which had been encapsulated. Okay. This is long. I'm just trying to read this with you because, I mean, trying to understand what they're doing here and what they're saying. Assembly of oligomeric pores as mechanism for membrane. I'm going to skip down here to let's do the discussion. I don't know that I can do the whole page. The results reported here point to a possible primary target for the cytotoxicity. Well, I mean, I was really kind of trying to see what it was they were doing. I mean, I know some of this stuff. They're building to achieve vesicle resolution in the leakage assays. We incubated the different protein constructs with GUVs in which calcium had been encapsulated. Okay. Confocal microscopy images. GUVs filled with calcium, which is the green. And time-lapse images follow calcium efflux, a progressive decrease in fluorescent intensity from GUVs upon his tagged. Inhibitors. Lipid vesicles have been recently used to monitor interference of small molecules such as natural polyphenols in the amyloidogenesis of an acyclion on membranes. Thus, we tested the possible interference of the polyphenols EGCG, curcumin, resveratrol, and curcumin on vesicle leakage by RepA-WH1 pores. All these compounds exhibited some inhibitory effect on calcium release from LUVs, as elicited by RepA-WH1, MCherry ranging from 10%. I don't know how to even read those things. Therefore, two compounds with their best inhibitory properties were curcumin and the, we need to see what that is. Also known as polyphenol, under research for potential to affect human health and disease. Yeah, but it's found in green tea. There you go. Of course, PubMed. There you go. So, what does that say? We need to be drinking green tea, having quercetin, is that how you say that? Resveratrol and curcumin or turmeric. That's, I used the full turmeric. Anyways, okay. Therefore, the two compounds that were the best were the curcumin and the EGCG, which were explored in more detail. The interval between showed a linear response with concentration. The action of polyphenols on the leakage promoted RepA-WH1 in the GUV's membrane model was also tested. Both curcumin and the EGCG substantially decrease the fraction of vesicles that release calcium upon incubation with the prionoid, confirming the protective role of polyphenols on protein-promoted damage to membranes. Now, see, that's awesome. Since we had found, so there's things I know that these things, okay, I'll just go on. Since we found a preference for the soluble RepA-WH1 A31V M-cherry dimers over pre-assembled oligomers in binding and leaking lipid vesicles, we separately tested in the calcium release assay using LUVs the most active polyphenol, curcumin. I don't know if I say that right, and on the dimeric and oligomeric fractions of the protein. The result showed that quercetin, I'm saying it wrong, reduced the release of fluorophore from the vesicles by both dimers and oligomers by RepA-WH1 A31V M-cherry, although more efficiently up to twice, and however you say that, for the latter than for the former. This suggests that polyphenols are most probably both This suggests that polyphenols most probably both interfere. Most probably, what the heck? What kind of fucking language is that? Most probably both interfere. Who writes this stuff? With a process involved in membrane leakage, i.e., lipid-promoted structural transformation of Okay. This suggests that polyphenols most probably both interfere with a process involved in membrane leakage, i.e., a lipid-promoted structural transformation of dimers, and remodel protein aggregates yielding species less active towards membranes. Assembly of oligomeric pores as mechanism for membrane damage by RepA-WH1. The result... Sorry about that. That's lovely. Okay. My glasses are my eyes are getting blurry. Okay. Since we found a preference for RepA-WH1 A31V M-cherry dimers over pre-assembled oligomers, less active. Okay. This results, see, I completely lost my place, my mind. The results obtained with both LUVs and GUVs converge towards a scenario in which RepA-WH1 A31V M-cherry molecules, upon binding to lipid vesicles, cause their leakage, with a preference for APLs as promoters of protein amyloidogenesis. So they're telling you that this bacteria does this to you, but regarding the mechanism for such damage to the membrane, one possibility is the formation of protein pores because there's no lactic disruption of the vesicles, even an alteration in their appearance, as previously found for synuclein. I don't even know if I say that right. However, an alternative lipid extraction mechanism could not be excluded. Therefore, we aim to directly visualize the complex is assembled by RepA-WH1, a model of lipid membrane. After probing a number of different conditions, we succeeded in assembling oligomeric RepA-WH1 A31V M-cherry particular particles in monolayers made of PLs with the average composition found in E. coli, which were precasted on carbon-coated EM grids. Particles were negatively stained with a uranyl salt, and EM images were acquired under minimal radiation doses. RepA-WH1 particles were individually picked and classified, and 2D projections reconstructed following standard procedures. Protein oligomeric rings were readily visualized, albeit their shapes were heterogeneous. Heterogeneity, which suggest a dynamic nature for the pores, precluded the assignment of a unique number of subunits in their composition. The overall dimensions of the protein pores were 8 to 9 nanometers for the exterior of the rings and 2 to 3 nanometers for the internal channels. The average 2D projections are fully compatible with head-to-tail arranged RepA-WH1 monomers, which should proceed from dissociation of protein dimers upon binding to the effector lipids, as reported for RepA double-stranded DNA promoted RepA-WH1 amyloidogenesis. As building blocks of the channels, these results directly support the mechanism responsible for vesicle leakage promoted by RepA-WH1 and most probably for triggering the toxicity of the prionoid in bacterial cells. It's the formation of pores at the membrane. So the results reported here point to a possible primary target for the cytotoxicity expressed by the RepA-WH1 Prionoid in E. coli, the bacterial internal membrane. These distinct mechanisms have been proposed for membrane disruption by amyloidogenic proteins, including the formation of pores by the assembly of protein monomers into ring-shaped oligomers, membrane thinning, and lipid detergent-like extraction. Since the last mechanism should imply the lysis of a significant fraction of the vesicles, which was not the case under our experimental conditions, and the second one seems incompatible with the observed persistence of aggregates bound to the membranes, the formation of pores by the protein insertion into the lipid bilayer as first proposed for, and I don't know, again, what the AB is, NA's, Sanuk, and I don't know how to, amyloid, something to do with amyloid, was the most likely mechanism for vesicle leakage induced by RepA-WH1. We've shown here that RepA-WH1 dimers, rather than pre-assembled oligomers, are the most active species in inducing leakage through bilayers, including acidic phospholipids. Furthermore, through E. coli lipids coating EM grids, RepA-WH1 is able to assemble as channel pores whose average dimensions match those for the pores built by other amyloidogenic proteins. Although under the number of RepA-WH1 subunits constituting the walls of the pores is variable with a mode of 5. Their densities are clearly compatible with protein monomers arranged head to tail rather than with dimers that are disposed head to head and related by twofold symmetry. This is relevant because, as for double-stranded DNA and seed-promoted amyloidogenesis, binding of RepA-WH1 to membranes consequently implies dimer location and pore assembly through monomeric intermediate. For distinct proteins and under different conditions, the conversion of membrane-bound and ring-shaped oligomeric pores into fibrils has been proposed as a plausible further step in amyloidogenesis. Interestingly, in the presence of effector double-stranded DNA, double-stranded DNA molecules or amyloid seeds, RepA-WH1 monomers assemble in vitro as hollow single or double helical nanotubes with internal diameter of 2.5. Sorry, I have to drink some water. 2.5. Okay. This is well within the range measured here for the RepA-WH1 channels in vitro, making feasible that bacterial prionoid could also build ring-shaped pores upon insertion into the membrane in vivo. Therefore, this could also be the case for the RepA-WH1 once assembled into the membrane. The dynamic nature of the pores could allow for the ring opening and subsequent helical extrusion of a free protein in an end enabling seeding and growth of an amyloid protofilament out of the membrane plane. The RepA-WH1 subunits constituting the pores, as my eyes blur, constituting the pores do not appear to be structurally distorted because the volume of the model of the RepA-WH1 monomer in its replication competent conformation fits well within the average electron density, whereas some degree of distortion was previously observed in the monomeric building blocks in RepA-WH1 amyloid fibers. This suggests that the possible transformation of ring pores into fibers could be linked in to a subsequent transition of RepA-WH1 from a pre-amyloidogenic to an amyloid structure. In addition, we show here that we assembled in the presence of lipids RepA-WH1 A31V M cherry protofilaments lack lateral association, unlike the mature RepA-WH1 amyloid fibers. Most probably, there's that word again, those words, due to steric hindrance imposed by the fused fluorescent reporter. So most probably due to steric hindrance imposed by the fused fluorescent reporter. Recent studies show that ring-shaped tubular cytotoxic oligomers of an A-synuclein, and I should learn what that A and those B are before I start reading all this. Anyways, like these described here, a RepA-WH1 also target membranes and lipid vesicles. But independent lines of evidence reinforce the significance of assembly of oligomeric structures for understanding the general mechanisms of amyloid cytotoxicity. The mammalian prion protein, as is expressed in E. coli, is not a fully infectious agent. It's only after being amplified, replicated, in vitro in the presence of cofactors when it acquires infectivity, and APLs together with nucleic acids are the most efficient of such cofactors. Similarly, the intrinsically disordered protein A-synuclein experiences conformational changes towards folding and or aggregation upon binding to nucleic acids or vesicles, including APLs. The findings presented here on promotion of RepA-WH1 assembly of APLs, together with the similar role reported for nucleic acid ligands, highlight that the bacterial prionoid, mammalian prion protein, and A-synuclein shared the same effectors in their pathways towards amyloidogenesis. More specifically, we've shown both LUVs and GUVs are the most efficient PLs in promoting RepA-WH1 aggregation. We could not ascertain the fine structure of these aggregates, but as recently shown for the amyloid, amyloid beta, peptide, and the liposomes, there may exist an inverse correlation between the order and the size of the RepA-WH1 fibers and the dimensions of the lipid vesicles. Interestingly, in E. coli, APLs are preferentially located in the inner membrane, both at the poles and the division septum, where they contribute to the selective location of specific proteins. In this way, PG and Cl could guide RepA-WH1 towards the bacterial inner membrane as the initial triggering stage in amyloid cytotoxicity, underlying the capacity of the minimal system made of RepA-WH1 and APL vesicles to its model physiologically relevant events for amyloidosis. Cl has been found to be required for mitochondrial membrane leakage, A-synuclein, and tau. Although the focus of the present study was to reconstruct in cytomimetic vesicles a possible route for RepA-WH1 toxicity in bacteria, it would be certainly interesting to explore if this bacteria prionoid would also provide more direct information on the role of membranes in human protein misfolding diseases. In this sense, the inclusion of the vesicles of cholesterol or gangliosides, which have a central role in eukaryotic membrane targeting by amyloids, thus should be the subject of future research. This work also addresses a direct comparison between the amyloidogenesis of RepA-WH1 A31V as it has been studied so far in vitro, and His and M. Cherry tagged form as studied in vivo, leading to the characterization of RepA-WH1 as the first synthetic bacterial prionoid. There you have it, right? The first synthetic, although according to CD spectroscopy, RepA-WH1 and M. Cherry behave as essentially independent folding units. Our data also indicate that the fusion to M. Cherry enhances the pro-amyloidogenic potential of RepA-WH1 into the full-length RepA and free energy change linked to the coupling of the sequence-specific recognition of DNA at replication origins, and protein conformational remodeling is naturally used to enable replication initiation. When fused to M. Cherry rather than to the native C-terminal WH2 domain, RepA-WH1 channels such free energy change towards the population of monomeric and aggregation prone metastable folding intermediate that leads the pathway towards RepA-WH1 amyloidogenesis. The most natural functional amyloids found so far in bacteria are secreting extracellularly. Through secretion, bacteria would protect themselves from amyloid triggering damage to the inner membrane, as been documented in his work. Heterologous expression of E. coli of proteins involved in human disease usually eyes are blurring, usually leads to aggregation of monodipolar inclusion bodies, which exhibit some amyloid character but are mildly detrimental to bacterial fitness. By dynamically assembling diverse aggregated particles with distinct toxicities and amyloidogenesis, the RepA-WH1 Prionoid fulfills in bacteria the role as a proteinopathic agent better than the heterologous expression of the other ones. The work presented here constitutes a proof of concept of the feasibility of engineering a minimal cytomimetic fully synthetic system to model the targeting and leakage of membranes by disease-related amyloids. Finally, we've shown that this system is amenable to screen for small molecules such as natural polyphenols with a broad spectrum against the plethora of amyloid. So we have, their methods. I won't read through all this, but there you have it. And Mel says that, which means all vaccines, GMO, and fertilizers infectious. So they were built. They've been built synthetic bacteria that they have released into the environment. I'm assuming she's saying that this agent is already in us. But there we have it. Thanks for listening, guys. I'll put the link down below.